Hello and welcome to Nikolai's genetics lessons, and today I'm going to talk about population genetics. And here's a problem for two genes, one with alleles A1 and A2 at frequencies P1 and P2 respectively, and another gene has alleles B1, B2 and B3 at frequencies Q1, Q2 and Q3. What are the expected frequencies of gametes with linkage equilibrium, assuming that P1 is 0.3, Q1 is 0.2, and Q2 is 0.3. What is the expected genotype frequency of A1, A1, B1, and B3 genotype at Hardy-Weinberg equilibrium? So if you know how to solve this problem, you may stop video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. This problem is a little bit tricky one, because here we have one locus with two alleles and another locus with three alleles. So one locus for the gene A with two alleles, A1 and A2, and another locus with three alleles, that is B1, B2 and B3. So in order to solve this problem, we can use Hardy-Weinberg equilibrium, that is equal to binomial expansion, but also because the second locus has three alleles, we also need to know what is a trinomial expansion. So Hardy-Weinberg formula is equivalent of binomial expansion. So for example, if we have alleles A and B, our initial formula look like this. And when we expand this formula, we are going to get A squared plus 2AB plus B squared. And this part we call Hardy-Weinberg equilibrium when we have two alleles, allele A and B, and diploid organism. So squared here stands for the diploid organism. If we would have triploid or tetraploid, we would put 3 or 4 here respectively. And for the second locus, we have three alleles, allele B1, B2 and B3. So we have to use modified formula or trinomial expansion, and this is going to be allele A plus allele B plus allele C also squared, because once again we have diploid organisms here. So our expansion would look like this: A squared plus B squared plus C squared plus 2AB plus 2AC plus 2CB. For your convenience and better understanding, I would put names of the alleles on top of each letters in our trinomial expansion. So let A would stand for B1, so this is going to be B1, and B would stand for the frequency of the allele B2, and C would stand for the frequency of the allele B3. So here we would have B1 and B1. So in order to get A squared, we have to get two alleles B1. So B1 multiplied by B1 would be A squared. I don't know if it is convenient for you to understand. We can use instead of ABC B1, B2 and B3, and in this case we would get here B1 squared, but I think it's also would be easy for you to understand if I use formulas that you get used to in your mass class. So for the B squared, we would have allele B2 and B2, and C squared would be allele B3 and B3. And here we would have B1 and B2 genotype, and here we would have frequency of the B1 and B3 genotype, and B3 and B2 genotype here. So now we can solve our problem. According to our problem, when we have a locus with two alleles, so one gene with two alleles, and allele is just a form of the same gene, but one can be different from the other, even in one nucleotide. So we would consider this two variants of the same gene. So we have two variants of the same gene that we call A1 and A2, and we know that frequency is P1 and P2, but only P1 frequency is given. And we know that P1 frequency stands for the allele A1. And we are looking for genotype that is A1, A1. So this is enough information for us. According to our problem, A squared represent frequency of the genotype that is A1, A1. And here would be genotype A1, A2, and B squared stands for the genotype A2, A2. So what we have to find? We have to find the genotype that is here, A1, A1, that is represented by A squared, and we know the frequency that is here, that is 0.3. So what we have to do, we have to multiply 0.3 by 0.3, and the frequency of this genotype would be 0.09. So this is going to be frequency of the A1, A1, or A squared in our formula that we call Hardy-Weinberg formula. And as for the second locus that has three alleles, B1, B2, and B3, we are going to use trinomial expansion. So this is variant of the Hardy-Weinberg formula for three alleles. So we have frequencies Q1, Q2, and Q3. And this frequencies is Q1 0.2 and Q2 0.3. And frequency for the Q3 is not given. But as you see, we are looking for the genotype that is B1 and B3. And we are given frequency of the Q1 that stands for the B1 and Q2 that stands for the B2. So let me underline this. So we know frequency of these two alleles, but frequency of the third allele is not given. How we are going to solve this problem? And this is very easy, because we know that total number of alleles for this locus is 3 alleles, and they represent 100%. So if we know the frequency of the two alleles, it's very easy to find the frequency of the second allele, because this is going to be as follows: B1 plus B2 plus B3 would equal to 100%. Or we also can say 1, because here all the numbers given on the scale between 0 and 1. And if we know the frequency of the B1, that is going to be 0.2, so frequency 0.2, and B2 is 0.3. It's easy to say the frequency of the B3 this is going to be 0.5. So if we add all these numbers, we are going to get 1 or 100%. In order to get 100%, we just have to multiply all these numbers by 100. So here we would get 20, 30, and 50. Now, in order to find frequency of this genotype B1, B3 in the gene pool for this locus, we have to use this part of the formula that stands for the frequency of the B1 and B3 alleles. And this part of the formula stands for the frequency of this genotype for this locus. So let's circle it, and this is going to be this part of the formula. So 2 multiplied by frequency of the allele B1, and this is frequency of the allele B1, that is 0.2. So 0.2, and by the frequency of the allele B3, that is 0.5. So multiplied by 0.5. And if we multiply all these numbers, we are going to get 0.2. So now we know the frequency for the first locus, and for the second also independent locus. And now, in order to find probability of this genotype, we have to multiply probability of the A1 genotype for locus 1, and this is 0.09. So let's write this down, 0.09, and we have to multiply by the frequency of the genotype for the gene B. This is going to be B1, B3 in diploid organism, and we have found that frequency of this genotype B1, B3 would be 0.2. So we put this frequency here, or probability. So now we just multiply these numbers, and the answer would be 0.018. Or in other words, on the scale where 1 equals to 100%, we just have to multiply this number by 100. If we need an answer in percentage form, so we have basically to move this decimal point 2 places to the right, and the answer would be 1.8%. So the probability of this genotype would be 1.8%. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.